Well, welcome everyone to our Nutrition 2020 session for today. Thank you for joining. My name is Marie Latulippe, and I'm a Senior Program Manager at ILSI North America. And as people start to join, we wanted to mention that this session was organized by ILSI North America in collaboration with the ASN Microbiology Research Interest Section. So we're very happy to have that collaboration. ILSI North America is a nonprofit scientific organization, and we focus on advancing food safety and nutrition science for the benefit of the public. We do not lobby, nor do we advocate for specific policies. Rather, we focus specifically on developing science. And we do that through a tripartite model, and you see the icon to the top right of your screen there. And in that, we bring together scientists from government, industry, and academia from both the US and from Canada. And then the intellectual contributions of these scientists from these multiple sectors are combined with pooled industry funding to allow us to advance science for confident decision-making by all sectors. And one of our key recent accomplishments is this seal of transparency from GuideStar in the platinum level. And if you're interested in learning more about what that means, please go ahead and visit our website, which is on the slide here, ilcna.org. And with that, I'd like to introduce to you our session chairs for today, Dr. Cindy Davis and Dr. Chris Cifelli. Dr. Cindy Davis is the Director of Grants and Extramural Activities in the Office of Dietary Supplements of NIH. And in this position, she actively engages and encourages partnerships with other NIH institutes and centers to facilitate the funding of grants that are of high relevance to ODS mission and goals. She's also actively involved in a number of government working groups that focus on the microbiome. Dr. Davis completed her postdoctoral training at the Laboratory of Experimental Carcinogenesis at NCI. She then joined Grand Forks Human Nutrition Research Center at USDA as a research nutritionist. And in 2000, she received a Presidential Early Career Award for Scientists and Engineers and was named the USDA Early Career Scientist. Dr. Davis also has numerous editorial roles with high-impact nutrition journals, and she's also an advisor to our ILSI North America Gut Microbiome Committee. And Dr. Davis is joined as a co-chair by Dr. Chris Cifelli. Dr. Cifelli serves as Vice President of Nutrition Research at the National Dairy Council. And in this position, Dr. Cifelli leads the Dairy and Public Health Research Platform, the goal of which is to provide scientific evidence on how dairy foods beneficially contribute to public health. And Dr. Cifelli is responsible for strategic planning, development, management, and communication of research on how dairy food consumption impacts chronic disease risk, childhood nutrition, digestive and bone health, as well as sustainable nutrition. And in addition, Dr. Cifelli leads NDC's NHANES research program. Dr. Cifelli is active in several professional organizations. He currently serves as the chair of the Science and Translation Committee for the International Scientific Association for Probiotics and Prebiotics, which many of you may know as ISAP. He's also the chair of the ILSI North America Gut Microbiome Committee and is on the board of directors for the Yogurt in Nutrition Initiative. So with that, I'd like to turn it over to Dr. Cifelli to kick off our session today.
Thanks, Marie. Cindy and I are super excited for this session, and we'd like to welcome you to it. We have three excellent speakers lined up. We'd like to take a second to acknowledge the planning team that helped develop the concept for today's session. This included the ILSI North America Gut Microbiome Committee, as well as Dr. David Sella, the chair of the Microbiology Research Interest Section. As Marie said, this is a partnership between the ILSI committee as well as that Microbiology Research Interest Section. Now, a little bit about the Gut Microbiome Committee of ILSI North America. Our mission, as it says on the screen, is to advance the science required to substantiate dietary modulation of the gut microbiome in ways that are conducive to health. And what we mean by this, and really what the overarching principle of the committee is to focus on three primary areas of work. the harmonization of research methods, understanding and shifting the field towards the use of best practices, and improving the link and collaboration between the fields of nutrition and gut microbiome research to help facilitate questions related to diet and gut health. And we realize that there's a lot to know between the gut health and the microbiome, so we are trying to link all that together. And we think that this session today will help us kind of get there. So without much further ado, I'd like to get into the session. We have, as I said, 3 excellent speakers lined up for today. The session will look at recent learnings about methodologies in diet characterization, sample collection, and data analysis. And the goal is to generate, of this, to kind of get you thinking about how we can generate research that will be examined collectively to answer critical public health and nutrition questions. And before introducing Dr. Slavin, I just would like to note that you as the audience can type your questions in the chat box at any time throughout the session. We did leave about 15 minutes at the end of this session to address those questions. So with that, I'd like to take a moment to introduce Dr. Joanne Slavin of the University of Minnesota. Dr. Slavin is a professor in the Department of Food Science and Nutrition and teaches advanced human nutrition there. With the help of her over 75 current and former graduate students, she has authored more than 300 scientific articles on dietary fiber, carbohydrates, whole grains, protein, snacking, and the role of diet in disease prevention. Dr. Slavin was a member of the 2010 Dietary Guidelines Advisory Committee, is a science communicator for the Institute of Food Technologists, and a member of numerous scientific societies, and has given more than 350 scientific presentations around the world. Dr. Slavin grew up on a dairy farm in Wisconsin, which she still owns with her sisters. She has a BS, MS, and PhD degrees from the University of Wisconsin-Madison and is also a registered dietitian. Dr. Slavin will address the characterization of diet in research and focus on the gut microbiome. I will now turn it over to you, Dr. Slavin. Thank you.
Thank you, Chris. I appreciate that so much. Okay, perfect. Thanks so much. I really appreciate the opportunity to present on this very important topic. So thanks for the introduction, Chris, and for the committee at ILSI for allowing me to give this talk today. Next slide. I just want to thank all the people that have supported my research over the many years that I've been in it, and all of my acknowledgments and competing interests are on this slide. Next slide. The outline today, we're going to first talk about what we know most about what affects the microbiota, and that is dietary fiber, and then all the data we have on other dietary exposures. We see a really weak link that we'll talk about, unfortunately, and some of it may be just because we have an inability to control dietary exposure or measure it very well. And then I want to end up on some of the best practices for dietary exposures and designing dietary interventions in this area. Next slide. Obviously, we eat all the time, so it's really hard to get after this and say, hey, what happens? So when we eat food, most of it gets digested, absorbed, and gets into our body. But there's a lot of undigested food components, mostly dietary fiber, that get down to the large intestine, and there's a fermentation process that happens. There are different products of that we'll talk about. And remember that this is always cycling back and forth. So this is a system that is very difficult to study just because there are things happening throughout the body. Next slide. What reaches the large intestine? If you're normal, and right now you just ate mostly fiber, oligosaccharides, lactulose, undigestible carbohydrates are most of what's going to get to the large intestine. But there's some malabsorption of foods, like 5% of ingested fat, 8% of ingested protein, and then unabsorbed vitamins, minerals, and phytochemicals all reach the large intestine. Next slide. I want to give you a little background on fiber. We know fiber is good for us. It's the marker of a healthy diet. All of these plant foods are recommended in our dietary guidance, but it's really different because rather than a vitamin or a mineral or a protein where we digest it or we get it absorbed, it's carbohydrates and lignin that escape digestion in the upper gastrointestinal tract, but can be fermented in the gut. So it's a very different concept. We believe it's a nutrient, but even that you can argue about. It is regulated. We know it's important. So on the nutrition facts panel right now, it's supposed to increase to 28 grams per day, and we have health claims. So we agree on fiber. There's a lot of things we agree on. Next slide. But you got to remember that all of our recommendations for intake of fiber, those DRIs we just talked about, are based on protection against coronary heart disease. So this is data that was used in the dietary reference intakes to come up with those recommendations. So they're not based on any type of effects in the large intestine. Next slide. Fiber is very complicated, and remember, it's going to spend 2 to 3 days in you in different ways. So it affects all parts of the digestive tract. It increases chewing, can slow down gastric emptying, can slow down the digestion and absorption of other nutrients. It affects different gut hormones, we know, but mostly what we're going to focus on today is fermentation, what's going on in the large intestine. So as these things get down there, products get produced that most of what we're interested in today are microbiota changes, but all of this, remember, as we've studied this, we have different things we know that are affected because of this large intestine fermentation. Next slide. So important thing that fibers are really different. So even though we have this category of fibers, and you might think of soluble versus insoluble, even beyond that, they're really different. So things like pectin and inulin are extensively fermented. So they basically all get broken down. And then we have things like purified cellulose that mostly survive all the way through. So only about 10% get broken down. So to say all fibers are the same makes no sense at all. But even fibers that get broken down, fermented, what do? They produce more fecal biomass because that bacterial mass gets increased, and that binds water. So feces are about 75% water, so that fiber or bacteria, both of those things, will increase fecal weight. Next slide. What happens? There's about 2 days, it's about 12 hours before stuff gets down to the large intestine. And then what's going to happen in the large intestine? Absorption of water, electrolytes, but this breakdown of anything that gets delivered to the large intestine. So it's mostly fiber, non-digestible carbohydrates, but there's some protein, phytochemicals, things like phytoestrogens, phenolic compounds. And so that breakdown is happening, and it's really hard to study. Appreciate that. Also, production of short-chain fatty acids, other bacterial metabolites that are happening, and this affects energy absorption and other things. And water feces, mostly water, but anything that doesn't get digested and absorbed, and a lot of bacteria. Next slide. So why did people get interested? I've been in fiber my whole professional life, and I've been very interested in fiber and the effects of eating different foods and what does that do in the large intestine. If you go back to this 2006 study that's been highly promoted, that gut microbes are associated with obesity. So in these studies, when you had people on different diets, what they found were differences in bacteroides, and that was actually related to a change in weight. So this really kind of springboarded everybody got into the microbiome revolution and said, hey, we want to do studies, we want to measure this. So it's amazing. If you look back to 2006, the number of publications significantly going up. Next slide. We mentioned all the things that happen with fermentation. I don't want to go into that in any more, but we know that like the colonocytes, the health of the colonocytes, really important, increasing mineral absorption. So things we hadn't even thought about. Do want to talk a little bit about the prebiotic concept because it's very similar to the fiber concept, but the stimulation of what are considered beneficial microbes. So let's talk a little more about that. Next slide. This was the ISAP 2017 publication trying to talk about prebiotics, not prebiotics, and how does fiber fit into this. So a couple of things I want you to get from the slide. If you go to the left side of the slide, these are things that are prebiotics. So obviously carbohydrates, readily fermentable fibers that are prebiotics. Obviously they fit, but they're also dietary fiber. Human milk oligosaccharides, we know that those are prebiotic. And then you'll see like conjugated linoleic acid, polyunsaturated fatty acids, which seems really odd. Why would fats have a role and be prebiotic? Also, all of the oligosaccharides, phenolic compounds, phytochemicals also being prebiotics. So appreciate that prebiotics overlap a lot with fiber, but it's a broader concept. And the definitions on the bottom, a substance that is selectively utilized by the host microorganisms conferring a health benefit. And it's been interesting watching the prebiotic go progressed in the literature, because what are those health benefits that we want to measure and how do we link that? Next slide. So I just wanted to mention everything affects laxation, everything affects the gut microbiota. So obviously fiber, really important, but all these other effects also will do that. So when you think about trying to power your study and coming up with a good study design, just remember that all these things are known to affect laxation and the gut microbiota, which makes it quite overwhelming to design studies. Next slide. All right, so how much fiber should you eat? We know that we are an omnivore, so we can go up to 80 grams per day. Recommended intakes are based on that cardiovascular disease slide I already showed you. So now 28 grams for a 2,000 calorie diet. Usual intakes only 15 grams per day. So we need to increase fiber, no disagreement on that. Lots of studies show we have the ability to tolerate different types of fiber really well, but some of the highly fermentable fibers like the fructooligosaccharides, there is good information that they tend to cause much more problems. They are poorly tolerated. So to say fiber is good for us, the right amount of fiber, plant food is good for us, but too much fiber can actually cause more problems. Next slide. I'm going to go past this because we know that microbes have many effects in the body besides short-chain fatty acids, producing vitamins, carcinogens. So that whole data on colon cancer, very important. Talked about calcium absorption and the immune system. So a lot of the interest in the microbiome has to do with the effect on the immune system. Next slide. Okay, so which are the dietary components that, can we agree that, where are we at here? What can we get agreement on? Most of the published studies are on fiber-resistant starches, but you'll see just a lot of disagreement in the regulatory field. So fermentation is not accepted as a physiological benefit in the FDA fiber definition, while it is in Canada. So the importance of fermentation as a benefit is argued. Lots of data on fiber, prebiotics, oligosaccharides, and I'm not getting into this at all today, but the whole probiotic field, fermented foods, yogurt, those effects, obviously, we know that those are going to also affect the gut microbiota. Next slide. So I wanted to just summarize what do we know and what are we going to recommend. So this was a recent meta-analysis, which was published in AJCN recently, 64 studies, 2009 participants. So dietary fiber and the gut microbiota, what do we know? So what can we agree on? Dietary fiber resulted in a higher abundance of bifidobacterium and lactobacillus, as well as fecal butyrate compared to placebo. Remember that most of the data they had was from the prebiotic world. So fructans, known prebiotic, galacto-oligosaccharides, known prebiotic, led to greater abundance of both bifidobacterium and lactobacillus compared to the control. So this is really the prebiotic literature that was focused within this review. What they did find, though, no differences between fiber intervention and comparators for alpha diversity, abundance of other pre-specified bacteria, or other short-chain fatty acid concentrations. So it's something that we say we know the most about. We still, when we try to do a meta-analysis, it's very hard to find a lot of studies, and it's hard to combine this. And this is basically the prebiotic field that is getting summarized here. Next slide. I'm very proud of my wonderful graduate students that are out doing wonderful things, and this is one of my former students, Katie Keckers, who published, we did a review paper on whole grain consumptions and changes in fecal microbiota. So trying to get all the studies that were out there and try to come up with some summary, did it very different types of grain, tried to get at that question. Whole grains generally modified the microbiota composition, but did so inconsistently across measurements of diversity and taxa. I think this is quite disappointing. We really were expecting a little more of a signal. And the part of the issue was the designs are all over the place. So people would use parallel designs where people were not switched over. They would use crossover designs. They might substitute one type of fiber or completely switch the diet. And the exposure either 3 to 12 weeks, so no consistency at all. And also how much whole grains were people actually eating, really difficult to figure out from these studies. So also the methods used to measure microbiota varied in their ability to resolve changes, and comparison of interventions using similar methods was lacking. So obviously, as people get excited about the field, a rush to do studies without really getting the methods nailed down at the beginning. Next slide. This is another one of my former wonderful students, Holly Willis, who worked together with me on an influence of diet interventions using whole plant foods on the gut microbiome. As you know, there's a shift from nutrients, fiber, to whole foods, plant foods. So what do we know about eating whole plant foods, and how does it change? We pretty much accept that plant foods drive microbial changes, but how strong is the evidence? And we really were expecting a little more of a signal here too. We went through thousands of studies, but in the end, trying to find well-controlled, well-defined human dietary interventions, there were 15 that netted out. And overall, there were no change, no effect on alpha diversity, and only modest effects on overall microbial composition. Interventions varied from 5 days to 24 weeks, and there really wasn't much detail on diet. These were the best that we could find on dietary control and the diet. What kind of diet were people eating, how much fiber, other things were in it. So there's no question that plant foods and dietary fiber is recommended, but it really can't be based on changes in the microbiome. Next slide. A couple other reviews I just wanted to focus on. These people took a really broad look at any dietary nutrients that affect the gut microbiota from 2005 to 2019. And in general, they basically found the same thing, that prebiotics that have been studied promote a range of beneficial bacteria and suppress potentially detrimental species. And I just wanted to highlight that because, what do we agree? What is a beneficial bacteria? So there's a lot of effort and a need to actually figure out what's beneficial for a normal person. And then, so obviously the biggest data, though, is in the prebiotic field, which includes dietary fiber. There is some data on quantity and type of fat that also would change bifido and lactobacillus. The protein did have some effects, but it was very mixed because of this, and little support for micronutrients having an effect except for vitamin D and polyphenols. Next slide. This is more of an epidemiological study and looking at in a group the adiposity phenotype study and using a measure of dietary quality rather than saying, hey, let's look at a specific nutrient, let's look at fiber, let's come up with a measure of diet quality. And they actually looked at different types of research or different types of metrics for diet quality. They found alpha diversity, according to the Shannon Index, increased by 1 to 2% across tertiles of diet index for diet quality, and there was the mean abundance of the phylum Actinobacteria was 13 to 19% lower with higher diet quality. So they believe diet quality is associated with fecal microbial alpha diversity, beta diversity, and several genera previously associated with human health. So interesting study, also one of my former students I am very proud of. Next slide. This is just a summary of some, the American Gut Project trying to answer nutrition questions. What do we know? And most of this data is very generalized, I'd say. So do we actually have good measures of how much fiber or how the diet is looked at? And in general, we did not, because it wasn't planned from the front end to do a good diet assessment. The diversity of microbes much more vast than originally thought. The thought, if I go from a non-Westernized diet, somewhat a loss of gut microbiome diversity, but hasn't always been shown in some of the more recent studies. More plant food intake have more diversity, also not some of the more controlled studies not supported by that. But remember, diversity has not been linked to a health outcome. It's been projected and suggested, but not done. We don't have that done yet. Next slide. So I want to there are two recent studies that really review articles that try to come up with best dietary practices. And some of the people that have helped sponsor this have really driven this, and I really appreciate it. So the first, David Clearfeld, an NIH USDA sponsored symposium to bring people together to provide full details and supplemental methods so studies can be replicated or analyzed in a systematic review. And a more recent research also showing considerations for the design and conduct of human gut microbiota intervention studies related to foods, also giving recommendations as best practices. Next slide. So I want to conclude. I really appreciate getting to lead off here and encourage people to take the diet seriously, make sure we analyze it. But most of the data we have, fiber, prebiotics, and probiotics. For other compounds to impact the gut bacteria, we must assume that they have to get to the gut to have that effect. The gut microbiota have lots of things that are affected by them. There's links between changes in the microbiota and health outcomes, but there really is no accepted healthy microbiota yet, and that's a real challenge in these studies. And then to just kind of send this home, standardized, validated methods to determine dietary exposure and standardized methods for gut bacteria are urgently needed for the field to move forward. As people rush to get into this research, actually having dietitians and other skilled nutrition professionals help design studies would do a lot to bring this whole area together. So I appreciate it, and thanks for having me.
Well, thank you very much, Dr. Slavin, for that wonderful start to this session. As a reminder, if people have specific questions, please use the chat function. We'll now turn to our next speaker. Our next speaker is Dr. Emily Boitman, who is an Earl Statman investigator in the Division of Cancer Epidemiology and Genetics at the National Cancer Institute. She has training in biochemistry and molecular biology and epidemiology from Michigan State University, the University of Michigan, and the University of Alabama at Birmingham. Her PhD dissertation focused on cruciferous vegetable intake, GST gene polymorphisms, and colorectal cancer among men in Shanghai, China. Completed in collaboration with the Shanghai China Institute and Vanderbilt Medical Center. Dr. Boitman joined the National Cancer Institute as a postdoctoral fellow and now as an investigator, where she conducts research focusing on the human microbiota and cancer risk. Dr. Boitman has also spent significant effort in evaluating methods for collecting, handling, and analyzing microbial data in order to apply these methods to her studies of cancer risk. Dr. Boitman will discuss the recent learnings related to sample collection and processing. It's our pleasure to invite you to be our next speaker.
All right. Well, thank you for that introduction. And I would like to start by thanking the organizers for inviting me to speak today. This clearly was not the plan for this session, but the conference and session organizers adapted quickly. And I'm very happy to share information with you all today regarding methods for collecting and processing samples of the human microbiota. And I would also like to thank Dr. Slavin for such a great introduction for what I'm going to present on today. The focus on methods has been a lot of my research because it's very important to collect and process samples in a way that facilitates pooling and meta-analysis in the future. To begin, I have no conflicts of interest to report. And I will today discuss 3 different areas. First, I will talk about collection methods that are available for human microbial sample collection. Second, I will discuss microbial sample laboratory handling. And then finally, I will conclude with some considerations for study implementation. So when one decides to start conducting a microbiome study, there's various aspects that need to be considered that may play a role in some of the differences that we've seen between studies, for example, of nutrition. So most of my research has focused on oral and fecal samples, but these samples have to be collected in some sort of media or in some way to preserve the specimen. And much of my research that I will discuss today has looked at how these sample collection methods may impact the microbial signature of the sample. From those samples, DNA needs to be extracted, and there's multiple methods available currently to extract the DNA, which may have an impact on the signature. Then there's also multiple different methods for sequencing, which depending on the method, it may also be difficult to pool the data because the data that's generated can be very distinct. Then the actual raw sequencing reads need to be processed in such a way so that we can identify the microbes or the functional capability of the microbes in the sample. And these methods may also play a role in some of the differences observed. And then finally, the data needs to be analyzed statistically. And it has been an issue looking at microbial data because it is very distinct from a lot of the standard statistical analyses, and new methods are currently being developed to analyze this data. To start, I will talk a little bit about different microbial sample collection methods. So when we've been doing this work, we have some specific criteria that we've looked at to identify a good collection method. The first is looking at technical reproducibility to see whether two aliquots from the same sample, how similar they are to each other. Next, we also look at stability. It's very unlikely that someone can have a participant provide a fecal sample on the spot. So it's very ideal to have a method that could be mailed from someone's home or could be brought into the clinic from the individual's home. So we've also evaluated the stability at room temperature for these different samples. And then finally, we look at concordance or accuracy. We want to compare these different methods to what's considered the gold standard. And so that is one other thing that we have looked at. And we often estimate all of these things using the intraclass correlation coefficient. For fecal collection methods, we've evaluated numerous methods, including fecal samples with no solution included with them, fecal samples preserved in RNA later, 70 or 95% ethanol. We've also tested two colorectal cancer screening methods, including the fecal occult blood test card and the fecal immunochemical test. So as I mentioned, when we look at these different factors, the reproducibility, the stability, and concordance or accuracy, we often use the intraclass correlation coefficient. And I'm going to orient you to the figure before I show the data, but an intraclass correlation coefficient ranges from 0 to 1, where 1 represents a very stable or a good metric, where 0 is the least stable. And so as we look across various factors, alpha diversity, beta diversity, and relative abundance for these different collection methods, we can see that some of the methods do not appear to be very stable at room temperature for this analysis. I'll focus right now just on the Shannon index, since it's difficult to look at all of those different metrics at the same time. But as you can see here, the no solution specimens, when stored at room temperature for 4 days, do not appear to be very stable. And similarly, the 70% ethanol did not appear to be very stable, whereas the other 4 methods did appear to have good stability at room temperature. So when we looked at stability, it seemed that no solution and 70% ethanol were not stable and are not ideal for sample collection if the sample has to stay at room temperature for a period of time. So to kind of give an overview of what I just mentioned, RNA later, 95% ethanol, the FOBT card, and the FIT test all do appear to be fairly stable using 16S RNA gene sequencing. We repeated this analysis for these methods using whole genome shotgun sequencing, and we found that 95% ethanol did not appear to be very stable at room temperature when using whole genome shotgun sequencing. However, the remaining methods, RNA later, the FOBT card, and the FIT test did appear to be stable. When we looked at comparability, the stable methods for 16S RNA gene sequencing generally appeared to preserve the rank order for the microbial metrics. So someone who had high alpha diversity in one method had high alpha diversity in the no solution samples, but the absolute values, the true alpha diversity estimates, did appear to differ. There was a lower concordance, a lower comparability for whole genome shotgun sequencing. So our recommendation from these studies is that we think it's very important to use one collection method for all study comparisons, because if all the cases in a study have used one method and all the controls use another method, you're very likely to see very distinct microbial characteristics, but it's very likely that it's related to the collection method. We also investigated stability and comparability for metabolomics, and unfortunately RNA later was unable to be used for metabolomics, so we did not evaluate RNA later. But what we found was 95% ethanol and then FOBT card appeared to be stable at room temperature for the metabolomic estimates, but the FIT test did not. And when we compared them to to the no solution samples, the 95% ethanol and FOBT card, again, generally preserved rank order for these metabolite levels, but overall, 95% ethanol detected the greatest number of metabolites. So it appears that 95% ethanol was best for metabolomics, but an FOBT card will also work. For oral specimens, I've conducted a number of different studies to compare collection methods, but also to look at how the temporal variability may impact sample size calculations for studies with outcomes. And so in this study, I collected samples from individuals at the National Cancer Institute over the course of 10 months. And at baseline, we collected saliva specimens in an OmniGene kit, as shown here. And at the second month, we collected another OmniGene specimen and also a scope mouthwash. Scope mouthwash has been used in a lot of large cohort studies, initially to look at human DNA, but now we have been using these to look at the microbial DNA in the mouth. At the 4th month, we collected another OmniGene specimen, and at the 6th month, we collected, also, in addition to the OmniGene, a non-ethanol mouthwash. In some of our populations, ethanol mouthwashes are not as common, so we wanted to see if a non-ethanol mouthwash would work for extracting microbial DNA. At the 8th month, we also included a saliva specimen preserved in Saccharomyces fixative, which is commonly used for sputum cytology. And again, at the last month, we looked at scope mouthwash and the OmniGene. So in this study, we were not only able to compare these sample collection methods to the OmniGene kit, but we were also able to look at OmniGene and scope mouthwash over time. So looking at the temporal variability, looking at the over time differences, we could see the percent variability explained in the different beta diversity matrices due to between subject differences, due to collection method differences, and due to the differences in visit or the temporal variability. And as you can see in this plot, a large majority of the variability in the beta diversity matrices was explained by between subject differences, whereas the collection method played some role and the visit did not appear to contribute a lot to the beta diversity variability. And then when we calculated ICCs for the variability over time, you can see that some of these ICCs, particularly for alpha diversity, were fairly high. However, the relative abundance of taxa did appear to have lower interclass correlation coefficients. So it looks like the oral microbiota is fairly stable over time, but the relative abundances of taxa are more variable. And the metrics with low stability over time will require larger sample sizes in order to have sufficient power to detect associations with an outcome for that metric. And in this manuscript, we present some power calculations based on the ICCs generated from these various metrics. We also in this study compared the different oral collection methods. And here are some peak away plots for the Bray-Curtis dissimilarity matrix, and comparing the OmniGene specimens, the scope mouthwash, the non-ethanol mouthwash, and the Saccharomyces fixative. Looking at principal coordinate 1 and 2, you can see that there does appear to be a cluster of the OmniGene specimens. And then looking at principal coordinate 1 and 3, and then 2 and 3, we can see a very distinct cluster of the samples in Saccharomyces fixative. So in additional analyses, we really observed that each collection method does appear to have an impact on the microbial signature. The two mouthwashes did appear to be fairly similar. However, they were not collected at the same visit, so we could not directly compare them. It does look like Saccharomyces at room temperature does not appear to be a good preservative for oral microbiota. And as with fecal samples, it's very important to use one collection method for all study comparisons. So moving on, I would like to talk a little bit more about, once you have the specimen, what to do with it. So here I'll focus on these parts of the pipeline of once you have your sample, you need to extract the DNA, sequence it, and then bioinformatically process the data. And to look at this, we conducted the Microbiome Quality Control Project. This was actually one of my first projects when I was a postdoc at NCI. And working with the steering committee, we recruited various laboratories across the United States and Canada in order to look at sources of variability in microbiome measurements across and within labs. And specifically, we wanted to look at handling, which incorporated DNA extraction, PCR amplification, and sequencing, and then also the bioinformatics. This was a volunteer effort, and we were able to recruit 15 different laboratories to participate for the handling portion and 9 laboratories to participate in the bioinformatics. The methods for this study are included here. We identified a set of specimens that could be used across all of the different laboratories. This included fresh stool samples, freeze-dried stool samples, samples from a chemostat or an artificial gut that grows liters of fecal sample, 2 artificial communities, and then negative controls. And from this set of specimens, we created a 96 tube sample set, which included 53 raw specimens. So these needed to be extracted in the laboratory. Then there were 41 centrally extracted DNA included in this set. So this could look at the downstream sequencing and bioinformatics. And then we also included the 2 negative controls. And laboratories could receive multiple sets of these 96 sample sets, because they could then also test some of their own methods within the laboratory. All of these sets were blinded and randomized, so the different samples did not always appear in the same location in the plate. And then each laboratory would then follow whatever procedures were standard in their laboratory, extract the DNA, PCR amplify and sequence the samples, and then they sent all of the raw data back to a central site, and that data was then blinded and randomized, and then that blinded and randomized raw sequencing data was sent to the bioinformatics laboratories to process it using any number of their own bioinformatics pipelines. And then all of that data was sent back to us, which was then unblinded, and we were able to do a data analysis using all of the laboratory data. So in the handling laboratories, we ended up having over 2,000 samples extracted and sequenced, which yielded about 215 million sequencing reads. Here are some of the methods that were used for handling in the various laboratories. Now, because this wasn't randomized and this was what people were using, there was less variability in some of the different variables than would have been nice. A majority of the laboratories used the Microbiota kit. Most of them were using the Earth Microbiome Project V4 primers. Almost all of the laboratories sequenced on the MiSeq, and most of them used paired-end sequencing. For bioinformatics, there were about 35,000 reads per sample, and then in the end, there were about a little less than 16,000 filtered samples. So here's a peek away plot of all of the MBQC samples. And you can see just by looking at the colors that in general, each sample type or each individual clusters somewhat together. However, within each sample, there is some significant variability. If we look specifically at the chemostat community, the majority of the chemostat is here in this box. However, some of it is also down near the bottom of the figure. So we were then able to use this data to investigate what caused some of this variability within each sample. And so we looked using Bray-Curtis, we were able to look at some of these different factors. Technical variation, so just looking at 2 identical samples, looking at bioinformatics, the sequencing, DNA extraction, and biological variation. And so to orient you to this graph before I show it, this is Bray-Curtis. So a value of 0 means it's very dissimilar or it's almost identical, whereas something up close to 1 means it's most dissimilar. We will focus on the human sample, the purple on the far left. And if you follow it, for technical variation, they're quite similar. For the same sample, just looking at duplicates, you can see that there isn't a lot of difference. The bioinformatics looks like there is some dissimilarity induced by bioinformatics, some by sequencing, and it increases for DNA extraction. And then finally, the largest variation is due to biological variation, which is what one would expect. So to look a little closer at the effect of DNA extraction, here is just one sample, and the different colors are the different DNA extraction kits. And you can see on this plot that the Microbiota kits do tend to cluster together, and each of the other methods do appear to cluster separately. So this really shows that there is a fairly large impact of DNA extraction on just one sample. So what overall we found from MBQC is that the largest experimental variability was due to DNA extraction method. There was a much smaller effect of bioinformatics. And then this MBQC study yielded a specimen set for intra-laboratory comparisons that includes quality control samples. And this is something we have used when we're testing a new laboratory that we're going to work with, that they can actually extract and sequence these samples, and then we can compare them back to the MBQC data. But there's a very important need for a larger scale randomized study, which can be used to evaluate the best DNA extraction method for multiple sample types, not just species, and for different sequencing techniques like whole genome shotgun sequencing. So now to conclude, I want to talk about some additional considerations for study implementation. I mentioned this just now with the MBQC, but it's really important to include quality control samples in your studies. One way to include quality control samples would be having a chemostat community like was used in MBQC. The image below is from Emma Allen Verco, who generated the chemostat communities for MBQC. And this is able to generate liters of an identical complex but natural type sample. People can also use sets of aliquots from volunteers. So just having generous donor samples to then have that same sample on every batch can also be used. It's also good to include artificial communities with dozens of known bacteria because you actually know the composition of that sample. Emma Allen Verco also generated the artificial communities for MBQC, but there are now also companies that generate these types of samples that can be used in the different batches. We also always include an extraction replicate of a study sample to just look at within a batch variability from an actual study participant. And then it's also very important to include blanks. In MBQC, when we looked at these artificial communities, in this plot, this shows differences by handling lab from one of these quality control samples. And you can just see that looking at the Bray-Curtis dissimilarity, that some laboratories were very distinct from one sample, that there was a lot of differences and some labs were very distinct. When we looked at bioinformatics, you can see that actually the y-axis is even different because there was a much lower impact of bioinformatics on the differences in the artificial communities. So by including these quality control samples, it can really help to evaluate intra and inter-laboratory differences. And this may facilitate pooling of data across studies and laboratories in the future. And then when we looked at the blanks in MBQC, this also yielded some important insights. You can see that one of the laboratories appeared to have some contamination of lactobacillus that wasn't observed in any of the other laboratories, but a number of the laboratories also appeared to have contamination in the blanks of Bacteroides. So this can tell you something of what's going on in your batches. In MBQC, at least one OTU was detected in almost all of the blank specimens. And the contamination may be from reagents, physical contamination from other samples in the laboratory, or other sources. And inclusion of blanks is really important in all batches. And additional blanks may be required for low biomass samples. And something that is really important to me is seeing the reporting of the results from blank samples. In a lot of manuscripts, they'll report that they included blanks, but they're never mentioned again. So some recommendations for microbiome studies going forward is that we think it's very important to collect and process negative controls at all points during sample collection, DNA extraction, and sequencing. And it's very important to use at least one or more of the positive controls, like a complex environmental sample, a chemostat community, or a mock community. In my studies, I include at least 2. It's also very important to report the DNA extraction methods in detail so that you can replicate these results. And there's also a need for the information of the positive and negative controls. It's also very important, given the impact of DNA extraction on the results, for large multi-institute or multi-site studies to utilize the same DNA extraction protocol. And I didn't go into this today, but there's also a need to demonstrate proof of life beyond sequencing for low biomass specimens. So overall, I just want to conclude by saying that there are many important aspects that need to be considered in a human microbiota study. I covered some of these today, but there's also other things that need to be considered. It's also very important to collect and process biological specimens and well-characterized cohorts using the best available methods, because population-based studies of the microbiota can likely provide important insight into the cause-effect relationship of the microbiota with health outcomes. And I think there's going to be many more exciting results in the years to come. So I need to thank all of the people that have contributed to the work that I presented today, in particular the steering committee from MBQC, all of the MBQC participants, my study participants from the oral microbiome study, and many other people. So I really appreciate the opportunity to present today, and I look forward to discussing questions at the end.
Thank you, Dr. Boitman. Our final speaker today, Dr. Long Nguyen, is an instructor in medicine and gastroenterology at the Massachusetts General Hospital and Harvard Medical School. He previously attended the University of California, Los Angeles, followed by graduate studies at Stanford University, where he obtained an MD in medicine and MS in epidemiology. He went on to complete a clinical and research fellowship in gastroenterology at Mass Gen, where he was mentored by Dr. Andrew T. Chan and Dr. Curtis Huttenhauer. He has since established his own independent research program focused on the dietary and lifestyle determinants of the microbiome, with a particular interest in sulfur metabolizing bacteria and their role in colorectal cancer. He is currently funded by American Gastroenterological Association Research Scholars Award. Dr. Nguyen will talk to us about the best practices for data analysis with some discovery from large HMPII study and an application to obesity and nutrition research. Dr. Nguyen.
Hi, thank you very much for the kind introduction, and an additional thank you to the conference organizing committee for the invitation to come and share what I've learned in the past half decade as an early stage investigator in the field of nutritional microbiome sciences. My thoughts are, of course, my own, but I'll be highlighting some team science produced in the laboratories of my mentors, Dr. Andrew Chan and Dr. Huttenhauer. I have no relevant disclosures for today's talk. The two big questions of microbial community analysis and associative microbiome studies is who's there and what are they doing? This is followed shortly thereafter by what does it all mean? There are properties of microbiome data that are, of course, not exclusive to the field, but still relatively unique for individuals like me who may have migrated from more traditional scientific domains such as epidemiology. They present challenges to our most common statistical assumptions and require extra care and consideration. Among them is the largely compositional nature of the data, where microbial or functional abundance is relative rather than absolute. Just like in nutrition, in which associations can be relatively challenging to attribute to nutrients, the foods that contain them, or the patterns that contain them, the tree of life in taxonomic order presents similar challenges. That is, are you interested in demonstrating differences at the phylum, genus, species, or strain level? There can be a high dynamic range of features, and with that, relative sparsity, in which samples with significant intra-individual personalization can be dominated by just a few taxa. And then there are unavoidable technical challenges that you heard a little bit about already, related to both the experimental and computational workflows. I would say there's very few Renaissance persons in this field who are expected to go from sample collection to extraction to sequencing to annotation and analysis. It's inevitable that you will be taking a data handoff from someone at some stage, so more than a cursory understanding of each step is required for reasons related to reproducibility. So here at the bottom of the slide, I've depicted a toy example in which the clade level abundance in sample/column 1 adds up to 1, just as it does in sample/column 3, with not very compelling or interesting results. Yet clear differences are observed at the higher level bug resolution. Building on that, and the previously alluded to statistical challenges, the compositional nature of the data can induce spurious correlation between features, i.e. bugs, gene families, enzymes, functional modules, in that an expansion of bug X is no doubt coming at the expense of non-X bugs. Technical challenges related to sequencing depth can require normalization to make more apples-to-apples comparisons. Relatedly, samples can be quite sparse, and thus an understanding of the upstream workflow can lend greater confidence to whether or not zero actually means zero. Is a zero a true zero representing absence, or does a zero in this case represent non-detection due to technical limitations? The hierarchical organization is especially challenged by imprecise language. We don't even yet have a universal definition of what a strain is, and yet it's critical to help define where your biology of interest may be occurring. This is particularly relevant because while core microbial gene elements responsible for housekeeping or universal functions are typically carried among wider members of a clade, genes and pathways responsible for more interesting niche functions may be contained only in a pangenome and thus highly strain specific. If I can bring this point home a little further, E. coli is perhaps the most well-known example of this, with strains that range from benign, like K12, to pathogenic, like EHEC, to even probiotic, like the Nissl strain. Comparable genomic plasticity is frequent among other common gut taxa, such as Bacteroides and Prevotella. As previously alluded to, the high dynamic range can introduce a central tendency to dispersion dependency, and this requires an appropriate statistical transformation to stabilize. And finally, the data is very noisy. It's easy to burn up statistical power, getting lost in the many features compared to the relatively smaller number of samples collected. By no means is this slide comprehensive, but often there are several large categories of modeling and association testing most suited to early pilot or smaller studies. Here on the left, there's multivariable association testing where a metadatum of interest, that is a patient characteristic, for example, is correlated to gut community profiles. Here in the middle, there's multi-omic association testing, which can help link two high-dimensional data sets, often linked by a given participant. The most common example of this being gut microbial profiles in stool and blood metabolites, though one can argue dietary data can be every bit as high-dimensional and omic as there is. And finally, there's ecological associations in which networks of co-occurrence and co-exclusion are investigated to reveal network-based trends. So diving back into multivariable association testing is the category of so-called omnibus testing. This works very well when the hypothesis being tested is an interest in global community alterations. That is there an association between some aspects of the entire community, such as diversity, with the metadata of interest, for example, age or biological sex? This procedure straightforwardly assesses whether overall patterns of microbial variation in a community associate with covariates by some significance model, with the most popular implementation being Permanova testing of dissimilarity matrices. Typically, this requires fewer number of tests, but it can be difficult to track down to individual features, that is, the microbes or enzymes that are most responsible for an association of interest. The input for such a procedure, we've already mentioned. The one caveat is that your measure of dissimilarity can greatly influence your results, and you heard a little bit about that from our prior speakers. Output typically comes in the form of statistical significance, which is a pretty low bar to clear with data this high dimensional, as well as the variation explained, often on the order of fractions or single percentages, given the overwhelming interpersonalized nature of the gut microbiome. One attractive feature of this type of analytical test is that comparatively small sample sizes can still be well powered compared to feature level testing. This represents an important complement and can offer a quantitative model to support qualitative data exploration. And I mention this because it's particularly germane to pilot or preliminary data generation. And here's an example of where even large-scale studies can rely on multivariable omnibus testing to offer sweeping, broad, but important observations. In work led by my former colleague published late last year in Nature, Dr. Lloyd Price, as just one element of a much larger project, performed large-scale omnibus testing in the Integrative Human Microbiome Project, colloquially called HMP2, an NIH-supported investigation of the multi-omic drivers of inflammatory bowel disease. While the number of subjects was low in the traditional sense, 132, the study's most distinguishing feature was the dense time series information that we had. What he was able to demonstrate just by using the Mantell test of correlation between matrices is rather impressive, in that between individuals, function reflects taxonomy, but they're not quite the same. Transcription reflects function, but that's not quite the same. Metabolites integrate information from all, including diet, which we treated as its own omic. Diet effects are modest overall in humans, but statistically significant. Within individuals, most omic types are similar between subjects. Transcripts in particular respond to perturbations around metagenomic function. And yet again, diet effects are modest, but among the most modifiable factors. Next, we'll dive into per feature testing. This type of testing is well suited to answer the question, are there any specific taxa or gene families or enzymes which are associated with the metadata of interest? Typically, this involves many more tests, but provides much more actionable information. Believe it or not, the most commonly used are correlation coefficients. There are some advantages to per feature testing. It's surprisingly effective for one, and it's relatively straightforward. However, it's characterized by a high false discovery rate, which requires type 1 error control for multiple testing. Also, it may provide a lack of context for interpretation in terms of biological plausibility and mechanistic insight. I'd be remiss if I did not mention what has now become a decade-long pursuit in our lab for how best to implement this at an epidemiologic scale. In pre-publication work by my colleague, Dr. Himmel Malik, we've developed what we're calling Maslin-2, or multivariable microbial association with linear models. It's available to download and use at the Hutton Howard Lab website. It's a multivariate statistical framework that finds associations between metadata and microbial community abundances or functions. And we've elected to base it on linear modeling. Using our benchmarking of synthetic data in real-world applications, we found that linear modeling offers the greatest compromise between high sensitivity with good FDR control. It can take the metadata form, almost any metadata form that you can think of. It can accept continuous data, such as age, weight, a dietary pattern score, as this audience may be investigating. It can take Boolean, true, false variables. It can take discrete factors, such as cohort groupings and phenotypes. Here we can see that in the same aforementioned IHMP or HMP2 dataset, we were able to fit a linear model adjusting for age, prior antibiotic use, participants' IBD diagnosis, and so-called dysbiosis to individual species and pathways. Beyond the scope of this talk is how we derived our so-called dysbiosis variable, but in brief, it represents our take on how large an excursion an individual had from a so-called typical microbiome. This method is described in detail in Lloyd Price et al. Nature 2019. So using Maslin-2, we were able to demonstrate that there's a robust and significant trade-off between the presence of facultative anaerobes, such as E. coli, and anaerobic short-chain fatty acid producers in the so-called dysbiotic Crohn's disease phenotype. So there in the teal box there, it's highlighted, and perhaps not the best visualization in this forum, is that anti-correlation in the context of other features, including, as you move across that row, a downregulation an upregulation of certain transcriptional pathways, despite a relative decrease in their DNA gene content. Another multivariable application relates to work that I helped lead, in which we derived a de novo dietary pattern using some tools straight out of Nutritional Epidemiology 101 to help address the long-held hypothesis that microbes responsible for dietary sulfur metabolism contribute to colorectal cancer carcinogenesis. The full details of this project were published earlier this year in Gastroenterology. So in a small pilot study nested within a much larger longitudinal cohort, the Health Professionals Follow-Up Study, we used reduced rank regression to generate a so-called sulfur microbial diet, a pattern of intake defined by food groups associated with increased abundance of 43 different sulfur metabolizing species of interest. We leveraged access to the smaller cohort with longitudinal stool sampling and dietary inventories to generate the sulfur microbial diet scoring system to us, which represents a data-driven, diet-based prediction for how much of these suspected bad bacteria a given individual may harbor. We found that this dietary pattern was characterized by food groups already known to be associated with colorectal cancer risk, including higher intake of processed meats and liquor, and lower intake of legumes and vegetables. Depicted below are the 8 food groups that most strongly contributed to sulfur microbial diet scores. After having this pilot data in hand, we then returned to our much larger, long-running observational cohort to generate sulfur microbial diet scores in all 50,000 individuals in the HPFS. And we were able to demonstrate that long-term adherence to this so-called sulfur microbial diet was associated with increased risk for distal colon and rectal cancers. So this to us offered first of its kind evidence linking targeted microbiome discovery, diet, and colorectal cancer. We feel that while the results themselves are meritorious and worthy of deeper exploration, perhaps our most important contribution with this project was to offer to others a methodological framework for extending lessons learned in pilot-sized nested controls to investigations at a population scale. Some tips and best practices for pilot investigations. Everything begins and ends at reducing dimensionality. So that may begin with enlisting the help of your local biostatistician for appropriate power calculations. That can include a deep dive of the clinical and experimental literature with your anticipated effect size and an understanding if the biology of interest is niche or more shared widely. It's not quite ready to be made into a blanket statement, but often longitudinal/repeated sampling is better. You'll want to choose carefully an appropriate statistical method. While beyond the scope of today's talk, using embedding methods for reducing similarity or dissimilarity input data, such as orthogonal feature reduction or an a priori gene or bug curation, such as we did with the sulfur project, is going to be important. You'll want to consciously decide on the number of dimensions to retain. So with that, I want to give thanks to the ASN, ILSI, and the Nutrition 2020 Organizing Committee for allowing an early stage investigator to share a forum with this esteemed panel. I want to thank my panel, my panel and my mentors. Software is freely available at the Hutt and Hauer website, and I definitely don't want to skip on the opportunity to recruit from this very talented virtual audience. Our lab's open for business, and we're looking for trainees at all levels. We pride ourselves on transparency, mentorship, and graduated autonomy. We pay a living wage in Boston, and we're particularly keen to work and train underrepresented minorities and first-generation scientists and health professionals like myself. We can teach anyone with the underlying motivation to join our ranks, so please write to me if you're interested.